What's happening, guys? Arouge, and today we're talking about cellular respiration, and this is the process of how we get ATP. This is how we get that energy molecule. Before we talk about the actual process of how we get ATP from glucose, we need to go over the parts of the mitochondria. And just like we went over the chloroplast, there are a few parts that I want you guys to know of the mitochondria. The first one is the mitochondria actually has two membranes. The first one is this outer layer, and this has a very difficult name. We call this the outer membrane. Okay? If we look inside, we have these folds right here. This would be the inner membrane. Okay? And if we look at within those folds, we have a fluid right here. And this fluid inside of the mitochondria is named after a really awesome movie we call The Matrix. Okay, so these are the basic parts of the mitochondria that we need to know. The outer membrane, we have the inner membrane, and lastly we have this fluid inside that is the matrix. Okay, go ahead and give that part a quick sketch in your notebooks. Okay. So if you look at cellular respiration, if we look at the products and the reactants and the products of cellular respiration, well, think about it. What do? We eat. And what do we eat? Glucose. We eat glucose, which is C6H12O6, and we breathe oxygen. Okay? So these are the reactants. What are the products of cellular respiration? Well, we breathe out carbon dioxide. Yes, our cells actually make some water, and we're also going to make some ATP. Now remember, when we went over photosynthesis, I said 3 things go in and 2 things come out. This is the exact opposite. 2 things are coming in. We have glucose and oxygen, and 3 things are going to come out. We have carbon dioxide, water, and we're actually going to make ATP. This is the exact opposite of photosynthesis. Photosynthesis took in carbon dioxide and water, and it made, with sunlight, okay, so this arrow, we'll say this arrow represents sunlight. Okay, carbon dioxide and water in the presence of sunlight yields glucose and oxygen. So these are actually exact opposites. The reactants for cellular respiration, those are the products of photosynthesis, and the products of cellular respiration are the reactants of photosynthesis. Okay, so they are exact opposite processes. One difference, though, is photosynthesis is using energy from the sun, and that energy from the sun is getting stored in glucose. In cellular respiration, we're taking glucose and we're actually breaking it, we're splitting it apart, and the energy that was stored in glucose is getting transferred to ATP. And at the very end of this, I'll show you why that part is important. If glucose already has energy in it, stored in it, why do we have to transfer that energy to ATP? And I'll show you that at the end of the lecture. Okay? So there's 3 steps for cellular respiration. The first step is glycolysis, and if you actually want to break that word down, glyco is referring to glucose, and lysis, you should already know. Lysis means to split or break. So in glycolysis, we're actually splitting glucose. What's made in glycolysis goes to another cycle, and that's called the Krebs cycle. It's actually named from, that's the gentleman's last name who discovered the cycle. And the very final part of cellular respiration is the electron transport chain. Okay, so let's go ahead and take a look at these each of these steps a little bit closer. So glycolysis. Glycolysis occurs in the cytoplasm. So if we're looking at a cell, it's occurring in this jelly substance. Okay, we're not using the mitochondria, not yet. Okay, so we're not using these little bad boys yet. Glycolysis occurs in the cytoplasm. Now, what's going to happen? Glycolysis is going to take glucose. We'll say this is my glucose molecule, and it's going to split it into two 3-carbon molecules called pyruvate. Okay, so if you notice, how many carbons does glucose have? 1, 2, 3, 4, 5, 6. That glucose is going to get split into two 3-carbon molecules called pyruvate. And this whole process is actually going to make a little bit of ATP. It's going to make 4 ATP molecules. But just like if you ever heard the phrase, if you want to make money, you got to spend money. Well, in glycolysis, we made 4 ATP, but it cost 2 ATP to start. So here's a potential test question for you. If I were to ask you how many ATP molecules were net, or was there a net gain of, your response would be 2. Because it made 4 molecules of ATP, but it cost 2 to start the whole process. So a net gain of only 2. Now, there's also going to be another molecule that's made, and it's going to be called NADH. So what's going to happen is there's going to be a molecule called NAD+. And through this whole process, NAD+ is actually going to take an electron from glucose, and then that's going to be converted to NADH. Okay, and the next lecture that we talk about, well, I'll explain why this is important, why we have NAD+ that gets converted to NADH after it grabs an electron. We'll talk about that at a later time. But let's go to the next step. The next step is called the Krebs cycle. And in the Krebs cycle, this one is actually occurring in the mitochondrial matrix. Okay, so it's occurring in this this fluid right here. Okay, pyruvate. Okay, so that those two 3-carbon molecules that were split from glucose, they're going to get further converted into a molecule called acetyl-CoA. Okay, now a couple things are going to get made in the Krebs cycle. We're going to get 2 more ATP, actually. We're going to make 2 more ATP. We're going to make more NADH, which is carrying electrons. And we're going to get another molecule called FADH2. That's basically another molecule carrying electrons, carrying energy. Okay. And the final part of cellular respiration is called the electron transport chain. So essentially, this whole process of respiration, a big concept of it is it's the transfer of electrons. Okay, so we transferred electrons from glucose to NAD+ to make NADH. More electrons get transferred to make more NADH and some FADH2. And then it goes to the electron transport chain. And what that is, that's a series of proteins in the cell membrane of the mitochondria. And basically these proteins are just transferring the electrons, okay? Now, basically these electrons are going to go through this whole series of transport proteins, and oxygen is actually going to be the last one to grab an electron. After oxygen binds this electron, it's actually going to form water, okay? So that's where water comes in. If you go back to the equation, I said we actually produce a little bit of water in our cells, and it's because O2 is binding, oxygen is binding with these protons that are made while these electrons are getting passed along. And in the whole process, we actually make 32 to 34 ATP molecules, okay? Now, this is what I want you guys to sketch in your notes. The first step we have is glycolysis, and that takes place in the cytoplasm. What glycolysis uses, it's going to use oxygen. It does not require, I apologize. It's going to use glucose. It does not require oxygen, and it's going to use 2 ATPs. It's going to make 2 molecules of pyruvate. It's going to make 4 ATPs, but it costs 2, remember? So we only get 2 total, and it's going to make 2 NADH. The Krebs cycle takes place in the matrix of the mitochondria. It's going to use oxygen and pyruvate, and it's going to make acetyl-CoA, carbon dioxide, 2 more ATP, NADH, and FADH. Okay, and those are just electron carriers. Next, we go to the electron transport chain, or the ETC for short. This takes place in the inner membrane of the mitochondria. Oxygen is the final electron acceptor. Okay? And the electrons, or it's going to accept those electrons from NADH and FADH2. What it's going to produce, it's going to produce some water and about 32 to 34 ATP total. So if we add up all the ATP that we've made from one glucose molecule, we can say that one glucose yields about, we'll say about 36 to 38 ATP total. Okay? One glucose could yield about 36 to 38 ATP. Now, why are we going through this whole process? When I said that there's already a lot of energy stored in glucose. Well, here's why. I had mentioned, I compared using ATP to similar to quarters at an arcade. If you want the arcade game to go, if you want to play the X-Men game, you have to have quarters. Well, you can't use $1. Okay, so if you have $1, how are you going to use that $1 to play the video game? Well, you're going to have to put it through the change machine. And in the change machine, that's going to give you some quarters. Once you get the quarters, then you can play your game. Now, what does this look like in the cell? Instead of $1, we're starting off with glucose. Okay, there's energy stored in glucose, but it's not in a usable form. Just like there's value in $1, but it's not in a usable form for the video game. Okay, so that glucose molecule, instead of putting it through a change machine, it's going to go through the cell. Specifically, it's going to go through the cytoplasm first in glycolysis, and then it's going to go through the mitochondria. After it goes through the cytoplasm through glycolysis, the Krebs cycle, and the electron transport chain, then we get our ATP. We get about 36 to 38 ATPs total. Just like if I cash in $1, I get 4 quarters. Okay? After I have my 36 to 38 ATP from the glucose, now I can have my cellular functions. Okay? All right, that was it for today. I know I went a little bit quick, but I promise I will clarify everything in class tomorrow. Okay? And I'll see you guys next time. Bye.